If you've been paying attention to health news in recent years, you've probably seen the headlines. Ozempic may protect against Alzheimer's. Weight loss drugs could prevent dementia. A brain imaging story in the Washington Post went viral. Social media lit up. And suddenly, the drug class that had already changed how we treat obesity and diabetes was being talked about as a possible answer for one of the most feared diagnoses around, dementia. Then, earlier this year, much more quietly, came different news. The largest clinical trial ever conducted on these drugs in Alzheimer's disease patients came back negative. No benefit. Zip. So which story is true? Here's what I want to tell you today. Both are true, and understanding why requires thinking through this as a clinician would think through it. Not the headline way, not the hype way, but how do physicians think through it? My name is Dr. Estrada Bernard. I'm a neurosurgeon, and this is the Spine and Brain Care Knowledge Center. Let's talk through this together. First, a quick grounding. GLP-1 stands for glucagon-like peptide 1. These are drugs that mimic a hormone your gut naturally produces after you eat and enables you to feel full, satiated. They were originally developed for type 2 diabetes because they help control blood sugar. Then researchers discovered that they dramatically reduce appetite and body weight. And that's how semaglutide, the active ingredient in Ozempic and Wegovy, became a household name. They work remarkably well for what they were designed to do. People lose significant amounts of weight, blood sugar improves, cardiovascular outcomes improve. That's your heart disease issues. That includes your risk for heart attacks. Kidney disease progression slowed down. Very promising results. But here's where the Alzheimer's story starts. Those GLP-1 receptors, the docking sites the drug attaches to on the cells, they don't only exist in the pancreas where insulin is made. We've found them in the human brain, in the hippocampus, which is the brain's memory center, in the prefrontal cortex, which handles reasoning and decision-making, on the brain cells that run day-to-day operations, and on the immune cells that protect brain tissue. That fact alone is enough to make any neuroscientist ask, what happens when you activate those receptors? Maybe we can control the immune cells and tamp down brain inflammation. Or maybe we could stimulate the brain cells in the memory center. Very intriguing possibilities. Now I want you to think of the brain the way you think of any high-performance engine. It needs clean fuel, low inflammation or corrosion, and a good maintenance crew. Alzheimer's disease at its core is a failure of all three of these factors. You get abnormal protein deposits, which are called amyloid plaques, that gum up the works. You get inflammation that the brain's immune system has difficulty handling, can't quiet it down. You get a breakdown in how brain cells produce and use energy. And woven through all of it is something called brain insulin resistance, the brain's version of type 2 diabetes. Just like other organs in the body become resistant to the effect of insulin, the brain can become resistant as well. And that's what leads to this concept of brain insulin resistance. This was demonstrated powerfully in a 2012 study that examined the brain tissue from people who had died with Alzheimer's, even people who had never had diabetes. The hippocampal neurons, the hippocampal, remember, is that memory center. Those brain cells in that area showed the same kind of insulin signaling dysfunction that we see in diabetic tissues. And the severity of that dysfunction correlated directly with how bad their memory had been before they died. Now, GLP-1 drugs are specifically designed to address insulin resistance. They reduce inflammation. In animal models of Alzheimer's, they reduce those amyloid plaques that I referred to earlier. They reduce the tau, TAU protein tangles that choke neurons and preserve synaptic connections, the bridges between brain cells that allow them to talk to each other. So the biological argument was not just plausible, it's grounded. It's grounded in real science, not just wishful thinking. When researchers started digging into large healthcare databases, I'm talking millions of patients' records, they found something that on the surface looked remarkable. Patients with type 2 diabetes who were taking GLP-1 drugs like semaglutide appeared to develop Alzheimer's disease and related dementias at a significantly lower rate than patients taking other diabetes medications. Multiple studies, multiple countries, many patients, multiple research teams. The numbers range from 25% to 42% lower risk, depending on how the study was designed and which comparison group was used. That's not a small signal. That's a clinically meaningful difference. And it got attention for a very good reason. But, and this is the part that headlines usually skip, observational data and clinical trial data are not the same thing. They answer different questions. And let's talk about that. Here's an analogy. Imagine you notice that people who shop at Whole Foods seem to have lower rates of heart disease than people who shop at convenience stores. That's a real observation, but it doesn't mean the store is protecting their hearts. It's more likely that the people who shop at Whole Foods also exercise more, sleep more, have more income, see doctors more regularly, and make dozens of other health-supporting decisions. The store is a marker for lifestyle, but not the cause of the outcome. So just because you see better outcomes in people that go to a Whole Foods store doesn't mean that the Whole Food store of itself is making them have the better outcome. Something similar was happening in these research studies. The patients in those database studies who were taking GLP-1 drugs tended to be younger. They tended to have better access to health care. They were more likely to have their blood pressure, cholesterol, and blood sugar actively managed. All of these things independently lower dementia risk. The studies also lacked important information about key variables like the ApoE gene variant that's the strongest known genetic risk for Alzheimer's disease. The observational data is real. The signal is real. It just doesn't prove that the drug is causing the protection. To answer that question, you need a clinical trial. So, Novo Nordisk, the company that brought semaglutide to market, ran the trials. They were called EVOKE and EVOKE Plus. They are the largest, most rigorous studies ever conducted on GLP-1 drugs and their effect on Alzheimer's disease. Here's the setup. 3,808 patients across 566 research sites in 40 countries. Everyone enrolled had early Alzheimer's disease, either mild cognitive impairment or mild dementia, confirmed by biological markers, that is, tests that you can measure, meaning these weren't just people with memory complaints. They had actual evidence of amyloid protein buildup in the brain. Half got oral semaglutide daily, the other half got placebo, a drug that was neutral, didn't have any effect. They were followed for about 2 years. The primary measure was what is called CDR-SB, the Clinical Dementia Rating Sum of Boxes. It's the gold standard clinical tool for tracking how Alzheimer's progresses across memory, orientation, judgment, and daily function. It's the same tool used in the successful trials of the anti-amyloid antibody drugs. Now here it is. The result? No difference whatsoever. The patients on semaglutide declined at essentially the same rate as the patients on placebo. The p-values, that is, the statistical measure of whether a result could be due to chance, were 0.57 and 0.46 in the two trials. For reference, a result is considered significant when p is below 0.05, so this wasn't even close. Every secondary measure, functional ability, time to disease progression, quality of life, global cognition, was also negative. Now, I want to make sure we pause here because this is where the story gets really interesting, not just disappointing. There's some useful insight here. Here's the thing about the EVOKE trials that didn't make the headlines, and this is what you need to know. In a subset of about 200 patients, the researchers took samples of cerebrospinal fluid, the fluid that bathes the brain and the spinal cord. They took samples before and during the treatment. And in that CSF sub-study of the research, they found something unexpected. Semaglutide was doing something in the brain. They found that levels of p-tau, a form of the tau protein in the brain that marks Alzheimer's disease, it went down. Markers of neuroinflammation went down. A protein called neurogranin, which reflects how well synapses, remember those connections between brain cells, how well they're functioning, that went down. These are not trivial measurements. These are the same biomarkers that anti-amyloid drugs move when they're working in research studies. So the drug was engaging its biological targets and having a measurable effect, but it just wasn't helping patients clinically. How do we explain that? Why would a drug produce measurable biological changes in the brain and still fail to slow the disease process? The answer almost certainly is scale and timing. Did you get enough drug into the brain, and did you give it early enough for it to have an effect? Let me give you an example. Let's say you're imagining a house that has been quietly filling with moisture for 20 years. The damage to the house's structure, corrosion in the pipes, deterioration in the structure, that's been accumulating for decades. Now, imagine you walk in, open the window, let fresh air in, reduce the moisture level drops. That's real, that's measurable, but the structural damage doesn't reverse because you cracked one window open and reduced the moisture at that time. The damage has been done, and that's likely what's happening in this situation. The first problem is delivery. Semaglutide was designed to stay within the bloodstream. That's what makes one's weekly dosing possible. But that same chemical design makes it very poor at crossing into the blood-brain barrier. The brain has a barrier that selects what can go from the bloodstream into the brain, and that's called the blood-brain barrier. When researchers measured actual semaglutide concentration in the cerebrospinal fluid, they found levels about 200 times lower than in the blood. So the blood was screening it out. So the drug was barely reaching the tissue that it needed to treat. The second problem is timing. The patients in EVOKE already had established, confirmed Alzheimer's disease. By that point, years, probably decades of amyloid accumulation, inflammation of the brain cell loss, all of that had occurred. The upstream metabolic mechanisms that GLP-1 drugs address may no longer be the primary driving decline in a patient who has already had significant brain damage. So even if you improve the metabolic state, just like that house analogy that I gave you, if the structural damage has already been done, it's not going to be reversible. And I think this is a crucial distinction, and it leads directly to what the evidence actually means with respect to prevention. Here's the reframing that matters. The observational studies and the EVOKE trials are not studying the same thing, and that's why you have different results. The database studies captured patients with type 2 diabetes, people with active, measurable metabolic dysfunction, elevated inflammation, and insulin resistance. They were taking GLP drugs for years before any cognitive symptoms had appeared, before any dementia had set in. Now, the EVOKE trials enrolled patients who already had Alzheimer's disease. Prevention and treatment are fundamentally different problems in medicine. We know this from other diseases. Take statins, for example. They prevent heart attacks effectively in high-risk patients, but they do not reverse established coronary artery disease in a patient who has had 3 stents placed because of severe coronary artery disease. That doesn't mean statins don't work. It means they work at the right stage. The hypothesis that deserves serious investigation now is whether GLP-1 drugs, possibly in formulations that better penetrate the brain, as are currently in development, could protect the brains of people who have metabolic risk factors, elevated inflammatory markers, or early biological signs of Alzheimer's, but haven't yet developed the symptoms. That is, if you intervene in the real early stages when there's a sign that there might be a risk, but before the damage sets in, would that make a difference? That is a hypothesis that the current evidence supports. That trial has not yet been run. So let me be direct about what the evidence means in practical terms. If you're considering starting a GLP-1 drug specifically to prevent Alzheimer's, don't. There's no evidence yet that supports this use. No guideline that recommends it. And the EVOKE trial confirms these drugs don't treat established Alzheimer's disease. Starting Ozempic for dementia prevention is not evidence-based medicine right now. Now, if you have type 2 diabetes and you're already on a GLP-1 drug, the observational signal, while not definitive proof of causation, seems to be pointing in the right direction across multiple studies in multiple countries. Your drug is also protecting your heart, your kidneys, and your weight, and reducing your risk for heart attacks. There is no neurological reason to stop it, and some suggested reason to feel good about the possibilities. Talk to your doctor about whether it remains the right choice for your overall metabolic management. If you or someone you love has an Alzheimer's diagnosis, and you're hoping these drugs might help, I understand that hope. But the honest answer, based on the best available evidence right now, is that semaglutide at the current doses in the current formulations does not slow Alzheimer's progression in patients with established disease. The biology is not wrong. The drug delivery and timing appear to be the limiting factor. That's where the science needs to go next. That's where the research needs to follow. Now, I want to close with something broader. The story of GLP-1 drugs and Alzheimer's is not a story of a promising treatment that turned out to be fake. It's a story of legitimate biological hypothesis meeting the complexity of a disease that has defeated far more targeted interventions than this one. What the EVOKE trial taught us is that biological target engagement, actually moving the right proteins in the right direction, is necessary but not sufficient. Concentration levels are important. The drug needs to reach the brain in adequate concentrations. The intervention needs to happen at a stage when the disease is still reversible, and the population needs to be the right one. Patients whose brain health is actually being undermined by the metabolic pathways these drugs address. None of the knowledge gained is wasted. It points directly to what needs to happen next. Medicine advances through honest accounting of what the evidence shows, not through the headlines that get clicks, and not through premature dismissal either. When you understand how a trial is designed, what is measured, what it found, and what it couldn't test, you're equipped to make decisions that are actually yours to make based on facts rather than noise or hype. That's what this channel is here to do. I'm Dr. Estrada Bernard. If this was helpful, please subscribe. We're building a library here, one evidence-based episode at a time, to equip you to make the best decisions for yourself and your family. And if you have a question about this topic or anything else in neuroscience, drop it in the comments. I read them. And as always, make healthy choices, and I'll see you in the next episode.